In the largest sense, the brain is organized like a team. Within it, different areas work like members of that team. Each has its job. This racer's brainstem, for instance, awoke him this morning and will put him to sleep tonight. How does he remember each turn of the track? How does he recall the long stretch ahead? Memories are made possible by tissue that sits atop the brainstem. It allows him intense emotions. It allows him to remember. And covering it all, the outer layer of the brain, a wrinkled sheet of cells laid out in functional continents. In the back, a region that relays what the eyes see. Again, on the surface, but now at the top, a ribbon relays information about touch. The feel of the wind on the driver's face, the pressure of his hand on the wheel. And just ahead of that ribbon of brain, another whose job is to orchestrate movement, sending commands to the spinal cord, then along the arm to the hand, telling it to change gears. In front of it all is the driver's frontal lobe, part of his thinking, planning brain, that bundle of tissue which provides him with a strategy to win the race. No single area of the brain can win the race by itself. No single tissue can function without the help of the rest of the brain. The brainstem, the midbrain, and all those regions along the surface are in constant contact with each other, always active, even when the body sleeps. How do we know all this? Thanks in part to the fantastic tools that the modern-day brain detective uses to look inside the brain. The CAT scan penetrates the skull to X-ray the brain's structure, showing the location of actual brain tissue. The PET scan forms still pictures of the brain's activity. Its colors show which areas are most and least active. One of the newest tools for looking at the brain is based on an old and established technology, the EEG, or electroencephalogram, in which the brain's electrical activity is recorded from sites on the scalp. The new invention is a computer program. It translates those electrical signals onto an actual map of the brain. The squiggles on paper are transformed into colors.
Janet, now you're going to be looking at a checkerboard pattern repetitively flashed on this TV screen. I want you to keep your eyes focused on the center of the screen.
Okay.
With this device, we can see the electrical changes of a brain at work. The bright colors represent positive electrical charges. Jesse Salb, the inventor, is watching the brain process a visual image. The signal from the screen flashes into the eyes and then rushes through the brain like this. Slowed down, we can watch vision deciphered. First at the back of the brain in a burst of yellow and red. About 0.3 of a second later, we can observe activity at the top of her brain as she realizes what she's just seen. One of the most exciting recent developments in studying the functions of normal and injured brains involves combining neuroimaging techniques. Joining magnetic resonance imaging, or MRI, with computer technology allows images to be reconstructed into three-dimensional pictures. One of the pioneers in using this new method is Dr. Aaron Bigler at Brigham Young University.
Current magnetic resonance imaging technology allows us to acquire the images of the brain three-dimensionally. This permits a sectioning of the brain at any level or focusing on any region that we would like to focus on. It's a marvelous technology that allows us to view the brain in any plane. It's very easy to see what I'm talking about by looking at this image that we have on the screen right now. I will set this into motion and it will become evident what we can do. The head will move around, tilt back, and we will now expose the brain. We see the right hemisphere on the right-hand side, left hemisphere on the left-hand side. The image will now come back to the top of the skull. The head will rotate down, now move, look at the lateral surface of the brain, and then make these cuts all the way through.
Dr. Bigler's techniques are used to investigate many different pathologic states of the brain. This man had a severe brain injury from a 30-foot fall. Injured areas are shown in green. Enlargement of the internal cavities or ventricles is shown in brown. And the combined image helps researchers to understand the overall effect of the injury on behavior.
Prior to the advent of contemporary neuroimaging methods, we had no way of looking at the brain of a living individual short of operating on the head. Now we can look at all levels of brain and brain function using these contemporary neuroimaging methods. Great advancements have been made in diagnosing neurologic disorders, and we hope and anticipate that these methods will allow us to discover better treatments and to understand neurologic disease and disorder better.
We now know that there are important differences between the brains of the two basic groups that comprise humankind, males and females. The question of even minor statistical differences between men and women is a highly charged issue. Both sexes have strong feelings about the very idea, and the evidence is only beginning to emerge. Nevertheless, there are statistical differences. They exist, for example, in the senses, smell, hearing, and touch, as well as in hand preference. It's now thought that hand preference is affected by sex hormones during brain development. About 10% of males are left-handed, compared to only 4% of females. Left-handers actually respond differently to the world. They include a higher percentage of artists, architects, and athletes, and they suffer more learning disabilities like stuttering, autism, and dyslexia. How do left-handers develop differently, and what is the effect of the sex hormones? Every male fetus receives jolts of testosterone from the testes. Very possibly, left-handers receive more. This is testosterone. Does the extra dose slow brain development in the left hemisphere, shifting males from a right-handed pattern to a left-handed one? We don't know. But we do have evidence of the power of the sex hormones to shape the brain and the body. Rats, like humans, have divided brains. Using special techniques to freeze, slice, and measure the cortex, Professor Marion Diamond at the University of California at Berkeley has found structural differences between the two hemispheres. She has also found sex differences. In female brains, the hemisphere thickness is symmetrical. In male brains, the right hemisphere is thicker. To find out whether she could reverse this male-female pattern, Diamond manipulated the early hormonal environment.
We decided to take the ovaries out of the rats when they were first born and look at the right-left differences when the rats were young adults. And we found that the pattern had switched. Instead of being symmetrical, we found a larger right hemisphere in the females without ovaries, very similar to the pattern in the males, especially in the visual spatial area.
Diamond also removed the testes of the males at birth and found that the front two-thirds of the male brain switched, the left hemisphere being thicker than the right. In other experiments, rats were exposed to 3 different environments: a barren, impoverished environment, a standard colony, and an enriched environment, filled with toys and social contact. Could these environments actually have an effect on the size and shape of the cortex?
The results show that the enriched rat's cortex was thicker than that of the standard colony rat, and the impoverished rat's cortex was actually thinner than that of the standard colony rat. So in essence, with enrichment, the brain grows, and with impoverishment, it decreases in size, a basic use it or lose it paradigm. We find sex differences in the animals that have been exposed to the enriched environment. In the males, we find greater changes in their visual spatial areas. In the females, we find greater changes in their general sensory cortex.
By measuring the thickness of the cortex, Dr. Diamond can judge the effects of these environments. She has 2 consistent findings. Experience can change the cortex at any age, and there are sex differences in how the cortex responds.
We don't have too much hesitation in extrapolating our findings from rats to humans, because rat and human brains are made up of the same basic constituents. And with time, many of the findings in rats are duplicated in humans.
Recent evidence has suggested differences in the cerebral cortex of male versus female humans. The right and left brains of males are more asymmetrical, while in females, the right and left brains are more symmetrical. There is also evidence that enrichment produces changes in human brains similar to those seen in rats. Gender development is influenced by both biological and environmental factors. Caroline Smith is studying the effects of how we treat children on gender development. She asked mothers to play with children they have never met before.
He's 6 months, so he can't really sit up. So if you put him down on the floor, put a hand behind him to support him. But that's it. You can do what you like, where you like. 10 minutes.
Smith is analyzing the difference between mothers' reactions to girls and to boys.
What would you like to see? What would you like to see? Would you like to see that? Would you just like to say hello for a minute, would you? That's a funny noise, isn't it?
The toys the mothers offer the babies are influenced by the child's sex.
Hello. 1, 2, 3. smile. That's a nice smile. I'd like to sit on the floor for a minute. Can you crawl? Are you going to start to crawl?
Boys are encouraged to be active, to explore their environment.
Here's the ball. There. That's right, you're getting there. A little bit further. That's it. You've got it. That's right. You've got it. What can you do with it? Off we come.
But sometimes the children are disguised.
Nice. Look at this. Oh, my goodness me. My goodness me.
Jamie is a boy, but for the next person, he'll appear to be a girl.
Is it funny? It's funny being a girl, is it? Yeah.
His mother will hand him over as Jane.
There we are. Here's a little girl. Very pretty. Very pretty. Okay. Here's Jane. There we are. Jane. Oh, you are lovely, darling. Aren't you? Look at the lovely darling.
Smith finds that we talk more to girls. We often tell them they are pretty, and we encourage them to be less active than boys.
A little bit of yellow here.
So we do treat boys and girls differently.
She's smiling at you, Jane.
We offer them different experiences, encourage different abilities.
She is. She's smiling right at you.
But the question remains, are the different characteristics of males and females mainly the result of experience? Or are they built into the two-sided brain?
Oh, yes. Oh, sweetheart. You're such a pretty girl. Such a pretty girl. Oh, darling. Look.
The answer is probably both. Yet this knowledge tells us practically nothing about individual men and women. Our specialized two-sided brain ensures that we vary in our tastes and talents, skills and abilities, needs and wants. The Aborigines of Australia have survived the trackless wastes of the continent for centuries. To do so, they have developed extraordinary visuospatial skills, a feature of the right hemisphere. Has the combination of experience and evolution changed the way they think? Aboriginal children do not perform as well as white Australian children on conventional verbal tests. One psychologist, Judy Carnes, thought the tests might be ignoring the Aborigines' real skills.
The children, I've found, know a great deal more than I do, and a great deal more than most white Australian people. They take for granted that we possess most of the knowledge they have, and we don't. They think that a sense of direction is built into everybody, for instance. And also, the same thing applies to all their knowledge about the wildlife of their region. They don't really seem to think that any of their knowledge is special, and it is very much so.
Dr. Carnes believes that Aboriginal children use their visual and spatial memories more than white children. She invented a game to test her theory. The task, to remember the positions of a set of objects on a board. The man-made object should be easier for verbally oriented children to remember, because natural objects are not as easily described and remembered in words. Carnes used both natural and man-made objects.
All right, open your eyes and see.
The most difficult group of all to describe verbally, 12 stones. Philomena has 30 seconds to memorize the positions of the stones. She can take as long as she likes to put them back.
Very good. I will tell you, they're almost all in the right places. That one's right, that one's right, that one's right, that one's right. So is that and that and that. That is. These two should be swapped around. Do you think so? I've tested children between the ages of 6-something and about 16 years and a bit now. And they always perform better than white Australian children. Oh, and also the rate of superiority, if you like, stays about the same. They perform at about about 3 years ahead of the white Australian children, so that an Aboriginal child of 7 years would perform about as well as a 10-year-old white Australian child. Not quite as well, but it's about a 3-year difference. They also tend to perform these tasks in even ways in terms of tempo. They don't hurry in and put a few back and then slow down. They seem to perform at the same rate all the time. And they also don't mutter or mumble, which a lot of white Australian children have done while they were viewing the tasks. I think in efforts to learn the names, hoping that that would help them to remember where the items went.
The two groups of children use different strategies and perhaps different parts of their brains to solve the puzzles. The cortex, the wrinkled outer layer of the brain, orchestrates talents and skills. It's a network of nerve cells. Music, language, mathematics, problem solving, and face recognition all are activated here among the billions of neuronal connections. The two halves of the brain may look alike, but they're not. They're asymmetrical. They don't have identical skills and abilities. Each side has its own way of knowing the world and processing information. By and large, each side controls the opposite side of the body. When you move your right hand, the left side of the brain is at work. When you move your left hand, the right hemisphere is in control. Most people have a dominant hand, a sign of the brain's asymmetry. Most mental functions, like language and spatial skills, are generally housed in one hemisphere or the other. This division of responsibility makes evolutionary sense. Normally, both sides of the brain work in concert. A massive system of fibers called the corpus callosum carries impulses to and from each side.
Have a silver clip ready, please.
In the early 1960s, surgeons revived an old technique of cutting the corpus callosum to control massive seizures in some epileptics. The operation works. It also provides an opportunity to study the different properties of the two hemispheres. It's important to remember that all of these patients suffered from poorly controlled epilepsy. Therefore, some of the findings may reflect changes in brain organization secondary to the epilepsy. Vicky is a split brain patient. Because of her epilepsy, surgeons severed her corpus callosum. Now, her right hemisphere can no longer communicate directly with her left.
I knew what I wanted to wear, and I would open up my closet, get ready to take it out. My other hand would like just take control. It would reach in and get something that I wouldn't want at all. You know, and then I get, I don't know, frustrated, and I throw the one on the bed that I don't want. Instead of, a lot of times I can't even hang them back up.
This antagonistic behavior between hands occurs only right after split brain surgery. Vicky actually does very well in her daily activities, even though her left hemisphere is disconnected from her right. It takes special testing to reveal the different processing styles of the two hemispheres. Vicky is frequently tested at Cornell Medical Center by Dr. Michael Gazzaniga.
Okay, look right at the dot.
Okay.
The tests are designed to send different kinds of information to each hemisphere.
I simply want you to describe them to me. Just name the picture you see.
Okay.
Okay?
All
So
right.
get ready and look right at the dot. Fixate the dot. Look right at the dot.
Football. Shoe. A woman. A person.
What else?
A girl, that's all.
Okay, I tell you what. Open your eyes. Your left hand. Right what she was doing. Close your eyes and just let your left hand go.
Vicky's right hemisphere doesn't have the language to describe the whole picture, but it can express itself through her left hand.
Good. What did you write?
Skipping rope.
Huh?
Skipping rope.
Skipping rope? No, let me show you what you wrote there.
Telephone.
Yeah, what was she doing?
Talking on the telephone.
Remember that?
Yeah.
Good. Perfect.
Vicky saw the picture in her left visual field. The image was projected to the right side of both eyes. The visual information then traveled to her right hemisphere. It understood the picture, but could only say woman. Then she was instructed to write using her left hand. Her right hemisphere directed her hand to spell out the other feature of the picture. Dr. Gazzaniga asked what she had written. The left hemisphere tried to guess. It had not seen telephone, but it had heard the right hemisphere say woman. The left hemisphere then searched for a word that might be associated with the question of what the woman was doing, and she said skipping rope. When both hemispheres later saw the written word, the left, which had heard the right say woman, could now give a complete description, woman on the phone.
How about another one here? You're doing just perfect. Oh, this is a good one.
Huh. I don't know how.
Both hemispheres respond to the words. The left triggers her smile, the right makes her clap.
So what did you see that time?
Both sides receive the command at the same time, but the left side can communicate its awareness. While the whole brain normally participates in communication, most of what we think of as language depends on the left side of our brains. The area for language on the left side is more densely wired and even structured differently than the same area on the right. In 1861, Paul Broca discovered the left hemisphere's role in language. This brain changed the course of neuroscience. It belonged to a stroke patient nicknamed Ta. He was given that name because Ta was all he could say. After Ta died, Paul Broca examined his brain. Because Ta could not speak, Broca concluded the damaged part of the left hemisphere was responsible for language. It's now called Broca's area. Ta's brain was recently rediscovered in a Paris museum. Scientists x-rayed it with a CAT scan. The black area in the left hemisphere is where the damage occurred.
Buenos días.
We now know that Broca was largely right. Damage to Broca's area does disrupt language. Until his stroke, Charles Landry had a thriving legal practice. At age 45, a blood clot cut off circulation to part of his brain and put an end to his professional career. At the heart of practicing law is a facility for language. Landry's stroke affected both his ability to understand language and his ability to express himself.
Contingent remainder is sell it, sign it, deliver it, or anything subject to the contingency.
When we talk about language difficulties, we mean problems with either grammar or the choice of words. And if you were listening to him, someone might say, well, his grammar's all right, and even though he takes a long time to find the words, he tends to find them. His difficulties are really all mechanical ones. He's slow, he's got trouble getting going, but none of this is really language disturbance. Well, I think that problem gets resolved very quickly when we start to examine his understanding of language. Do you ever have any trouble with what I say?
No.
Understanding it?
No.
Well, suppose you wanted to communicate with a person who was in a distant city. What apparatus would you utilize?
Telephone.
Very good. All right. I deliberately made that sort of a curious sentence just to show how well you did. All right. Now let me ask you, do dogs fly?
No.
No. And how about this? Do submarines usually fly?
No.
Mm-hmm. How about a Zeppelin? Can a Zeppelin fly?
Yes.
All right. Okay. I couldn't make the question complicated enough in terms of the fanciness of the vocabulary to throw him off. He immediately understood. And yet, when I produced a sentence that most of us would have thought was about as easy an English phrase as you could create, he failed totally. You know what a leopard is?
Yeah.
You know what a lion is?
Yes.
Right. So the leopard was killed by the lion. Which animal died?
I don't know.
That's hard, is it?
No. I don't know. What animal died? I don't know.
You don't know what animal died.
Right.
Obviously, that's got to be an error in understanding the grammar. Now, there's a very easy way to understand it, because if you think of those sentences with the grammatical words removed and try to figure out their meaning, you would have the same problem.
Landry hears all the sounds of the grammatical words, but in his brain, the sentence is processed something like this. He understands only nouns and verbs.
This pattern of having this special kind of understanding difficulty, which is confined almost exclusively to these little words, to the grammatical words, and to the endings, is something that we find very frequently in the patient who's got damage to Broca's area. So the thing that's so perplexing in Mr. Landry is that he doesn't have this difficulty in spontaneous speech, he doesn't have this difficulty in repetition, but when we come to the comprehension tests, he has this difficulty in the most dramatic fashion.
Broca's discovery was important, but it wasn't long until other investigators discovered additional areas in the left hemisphere that contribute to language. In 1874, the German scientist Karl Wernicke identified a third language area in the left hemisphere. Broca's area in front plays a major role in speech production and grammar. The strip in the middle controls and coordinates movement, but Wernicke's area is involved both in the formation of what is said and in comprehension. When it's damaged, speech seems fluent but doesn't make sense. We now know how we process and repeat words. First, sound travels as nerve impulses to Wernicke's area, where it's analyzed, to Broca's area, where sounds are assembled into sequences, and finally to the motor cortex, which directs movement and sends signals to the speech muscles. Charles Landry's brain damage has produced difficulties that are fairly typical of patients with damage to Broca's area. From patients like him, we have learned a great deal about how the left hemisphere decodes, processes, and produces language.
Times I caught fish. Oh, boy.
Another way we're discovering how our most important organ works is by observing how it adjusts to serious injury. In certain cases, its power to heal itself is almost miraculous. Take hydrocephalus, an often fatal abnormality of infancy when the normal flow of spinal fluid out of the brain is blocked and pressure builds up inside the skull.
In the normal brain of a baby, the ventricles are that butterfly-shaped structure. On this brain of a hydrocephalic child, from the outside it looks roughly normal. It's not normal. But when you turn it round, these ventricles are enormously dilated, and the brain is only about half a centimeter thin.
In 1955, a valve and shunting procedure was developed. It permitted the release of fluid from the brain in a long bypass to the heart for recirculation. Since then, the valve implant operation has saved the lives of hundreds of thousands of hydrocephalic children. With no continuing trauma to their brain, many have become normal. In a few remarkable cases, some, like Sharon, have done even better.
I always thought she was about average, but now I'm beginning to wonder if she's probably more than average. She got every exam that she took. She didn't fail anything.
Even the English one?
Yes, and the English is the one that she's not too sure of. The chemistry, she was the only girl to get a GCE in her school. And in biology, she was the only one of two girls to get that GCE.
How can Sharon possibly be above average? What does her brain look like? Could it have assumed a normal shape? Sharon's consulting pediatrician, Dr. John Lorber in Sheffield, England, tried to find out. Sharon underwent a CAT scan to reveal the interior structure of her brain. In the normal brain, a CAT scan shows tissue completely filling the skull, with a shadow of the butterfly-shaped ventricles in the center. But in Sharon's scan, the image is extraordinarily different. The tissue of her brain is nearly flattened against the wall of her skull. How has she survived so well with such a distorted brain? Since a CAT scan cannot answer this question, Sharon was flown to Denmark for a cerebral blood flow scan. When she inhales xenon gas, the neurological detectives will be able to observe her brain's activity and may discover how she is making such brilliant use of her distorted brain tissue. The picture appears. It shows that activity in Sharon's brain is actually focused at the back, on the bottom of the screen, in the region normally reserved for vision. The normal brain, on the other hand, tends to distribute tasks evenly throughout. Neuroscientists use the word plastic to describe how function in Sharon's brain has been redistributed from one area to another. While not as complete an example of plasticity as Sharon, Nicole presents another remarkable case, illustrating redistribution of brain function following early hydrocephalus.
Nicole is a young lady that I've been following for a number of years, ever since she was a child. In infancy, she developed hydrocephalus and required shunting. As a consequence of the hydrocephalus, extensive areas of the brain were damaged, including most of the left hemisphere.
CAT scans of Nicole's brain show the extensive damage caused by hydrocephalus. In this view, the left side of her brain is shown on the right. The general expansion of the ventricles is more prominent on the left side of the brain. The large dark area in the left rear of the brain is a cystic formation resulting from hydrocephalus. It is filled with cerebrospinal fluid. There is no brain tissue there at all.
Traditionally, the left hemisphere is dedicated to language function. In Nicole's case, it's been extensively damaged, including the occipital area, the area that we see with. In her case, this has affected her ability to do visual perceptual tasks. She has difficulty seeing things and identifying objects. She has difficulty carrying out any type of visual motor task, movements with her hands, copying, using a pencil. But a very interesting and surprising thing happened early in Nicole's case, and that was that she developed normal language skills. Fast as you can. Great. Now do it with your other hand. The same thing. Okay.
Dr. Bigler's
Bring both your
evaluation
hands
shows
together
clear
and
difficulties
do it.
in Nicole's motor abilities.
Okay. Now try to do it where you just move this finger to your thumb, this finger to your thumb, and not move any of your other fingers, and do it both hands together simultaneously.
Nicole is unable to copy visual patterns directly in front of her. However, reading has developed normally.
I'd like you to read this for me if you would.
Okay, do all the good you can by all the means you can, in all the ways you can, at all the times you can, to all the people you can, as long as you ever can.
Very good.
Nicole's case, like that of Sharon, raises more questions than it answers. But both cases illustrate the unusual plasticity of the brain. Uninjured areas can take over functions normally regulated by the damaged areas. Nicole's case also shows the premium the brain seems to place on the ability to communicate through verbal language. Just how the brain accomplishes these feats is still unclear. It remains a challenge for neuroscientists.
An escalator, gate.
Vision is one of our most important senses. In order to understand how this sense works, let us go to the beginning of the visual pathway, the eyes. The eyes don't really see. they are mere sensors, beaming information to be deciphered and reassembled by at least 3 million brain cells. In most vertebrates, each waking second, the eyes send a billion items of information to the brain. But what do all these neurons do? And how do they allow us to see? First of all, the lenses of our eyes turn everything upside down. Then the visual information is beamed along the heavy fibers of the optic nerve. The first bend in the pathway is the optic chiasm, where some of the information from the left and right eyes crisscross. Then the pathways reach a major staging stop at the back of the head, a thin veneer of brain tissue. Here, at the primary visual cortex, also called the striate cortex, visual information begins its processing. At the University of California at Berkeley, a team of scientists led by Russell D. Valois and Roger Tattel set out to find just what information this primary visual cortex receives. This image was flashed before the eyes of a monkey. How would the nerve cells at the back of the brain encode the picture? Are visual images exposed onto the back of the brain the way images are exposed in a dark room onto photographic paper? Dr. D. Valois and colleagues came up with a surprising answer.
The technique that we used was to give the monkey minute quantities of radioactive sugar, which is taken up by the most active cells in the cortex. Then when we looked at the X-ray picture of the cortex, we could see which cells were most responsive to this pattern. And the amazing thing was the precision of this pattern that we saw in the cortex, where you can see all of the stripes and rings laid out beautifully on the cortical surface. We were really amazed at how precise the mapping was onto the cortex.
Photographs on the brain? Not quite. Without doubt, Russell D. Valois and his colleagues have shown that there is precision in the way individual nerve cells fire in response to what the eyes see. But this autoradiograph doesn't yet explain how those nerve cells actually decoded the image. To solve that mystery, Dr. David Hubel of Harvard has invested nearly 3 decades of research in pursuit of the answer. He and his colleague, Dr. Torsten Wiesel, made discoveries which won them the Nobel Prize. The brain surprised them, the way in which nerve cells of the primary visual cortex responded to an image flew in the face of their own intuition.
To start with, the cortex is a plate of cells a couple of millimeters thick and a foot or so in surface area. The part of the cortex that we're dealing with, the primary visual cortex, is a chunk of that, about the size of maybe a credit card. And here's the real thing. This is an actual brain of a macaque monkey. This is the primary visual cortex, spread out smoothly and with a tucked under part that we can't see. Here we've made a cut in the cortex, and to see what the actual cortex looks like under a microscope, we can walk into this region and look to the left, and you see on the next picture what that looks like. Here is the smooth surface of the primary visual cortex. This is the part that's tucked underneath, and now you begin to see that it's a layered structure. Some places the cells are packed tightly, other places they're looser. And underneath every square millimeter of cortex, which is about that much, you have something like 100,000 cells.
The researchers actually listened in to individual nerve cells firing in the anesthetized cat as they presented it with different visual images.
When we started working, Torsten and I, in the late 50s, we set up our first experiments, and they didn't go well because at the beginning we couldn't make the cells fire at all. We'd shine lights all over the screen, and nothing seemed to work. And rather by accident, one day, we were shining small spots, either white spots or black spots, onto the screen, and we found that the black dot seemed to be working in a way that at first we couldn't understand until we found that it was the process of slipping the piece of glass into the projector, which swept a line, a very faint, precise, narrow line across the retina. And every time we did that, we'd get a response. And even more than that, the line produced a response as it swept across the screen in one direction, but not in the reverse direction. Of course, that could have simply been an oddball cell. We didn't know whether we'd ever find another such cell. But after some weeks or months, it became pretty clear that most of the cells that we encountered in the visual cortex demanded just that kind of stimulus. Although from one cell to the next, the orientation varied. A number of other things differed. The line was the important thing.
Russell de Valois' findings do not necessarily contradict, but extend the findings of Hubel and Wiesel. They imply that some nerve cells are sensitive to the spatial frequencies of light, as in the subtle effects of sunshine on water, shadows spangling up on other shadows, a prismatic swirl of texture where edges do not seem to be the key factors.
It's very non-intuitive, but if you take a grading pattern and add it to another pattern, it still looks like just sort of a vague random pattern. But as you continue to add patterns of different periodicity, different variations across space, different orientations, pretty soon it builds up into a more and more complex pattern, in fact, one that you can recognize as an identifiable human face.
In the 19th century, Impressionist painters sensed that such spatial frequencies might help create and enrich imagery. Whole canvases emerged out of formless points. These seemingly random dots show how spatial frequencies can operate, how clear images are sustained out of a vibration of color and light. But David Hubel is not convinced by this theory of vision.
It might seem as a real surprise that the cells of the striate cortex have this preference for lines, because after all, it's obvious that we don't see lines all the time. We see objects. But if you think of it for a minute, you realize that objects themselves consist of light-dark contours. And what the visual cortex is doing is asking at any point in the visual world, is there a contour? And if so, what's the direction of that contour? So it really isn't lines that are being detected, it's contours and their directions. And that is probably sufficient information to tell you all you need to know when you look at a scene.
The primary visual cortex, this brain area the size of a credit card, has been studied extensively. But laws about the way it decodes vision are still not easy to lay down. While we may surmise that orientation, edge, and frequency detection are indeed roles performed by some of these cells, there are many other cells whose function is still unknown. Seeing is only a small part of perception. Deciphering, decoding the image of a well-camouflaged moth, for instance, requires pathways beyond the striate, or primary visual cortex. So too these random spots, bars, edges, and visual frequencies give us a sense of size and shape, but they tell us little about Dalmatians. Higher visual areas in your brain are now at work, a region of near unexplored brain territory to which Dr. Mortimer Mishkin has devoted much of his research.
The striate cortex is simply the first station in the cerebral hemispheres to receive information from the retina. And it looks as though the striate cortex in the back part of the brain sends information forward along two different pathways. One headed to the top part of the brain, to a region that we call the parietal lobe, and another headed toward the bottom part of the brain, toward a region that we call the ventral part of the temporal lobe.
That higher pathway, plunging in and out of the exterior cortex, makes a series of very specific visual computations. Its role is to tell us where things are in the visual field, to specify that the dog is in front of the mother and child, to establish that this couple faces the faraway lake. Clearly, this pathway helps us orient one object to another, a visual back and forth that continues as long as our eyes remain open. The lower pathway's function is possibly the most surprising of all. In a similar series of stages, it helps us explain what objects are. It tells us that this is a dog, this is a child, this is a man. As neuroscientists investigate our visual system, they find that our brain cells here are very, very specific. In the 1950s, some neuroscientists called them grandmother cells. The theory, which enjoyed a certain popularity, was that we might actually possess a single cell that lit up when our grandmothers came into view, and another cell for each and every person we ever saw. These cells were pictured as being almost miniatures of that person. However startling the theory, it now seems that these cells at the end of the lower pathway actually might be similar to grandmother cells.
Some of my colleagues working in Princeton and at Oxford University have recently shown that they can record from single neurons in part of the inferior temporal lobe, and these neurons will be activated by faces better than by any other kind of stimulus that they can present. Not only will they be activated by faces, but if the faces are distorted in certain ways, the neurons will not respond.
As we watch, we can hear single cells in the inferior temporal lobe of monkeys reacting to a single monkey face. The cell only fires if the face is at the right angle and is not distorted. What distinguishes these very specific cells from the so-called grandmother cells of the 1950s is that they rely on many other cells in the visual pathway to feed them detailed information. They're called cumulative cells. And so, grandmother is not merely embodied in a single cell. She's represented in our brains by a multitude of cells, some so simple that they merely analyze the bars and edges of her face, the visual frequencies of her complexion, and some so complex that in the lower pathway of vision, a single cell might actually light up when we see our own grandmothers, but only with help from the hundreds of thousands of cells that precede it. Utilizing modern brain research techniques, Dr. Leslie Ungerleider, a colleague of Dr. Mishkin at the National Institute of Mental Health, has been studying the process of human facial recognition.
Until recently, our understanding of how the brain codes visual information has come mainly from studying the activity of single neurons in the monkey brain, and also by looking at impairments in patients following brain damage. But now, with advances in the new technology of functional magnetic resonance imaging, we can look directly at activity in normal, healthy individuals' brains, and we can see how that activity changes as these human subjects are involved in various kinds of tasks. We're especially interested in what happens in the brain when people perceive, recognize, and recollect faces. Just as we had found in our monkey work, our imaging studies in humans indicate that there are specialized regions in the temporal lobe of the human brain that appear to be specialized for the processing of faces. When we see faces, these areas of the cortex become active. Imaging studies have also shown that beyond the visual cortex, there are areas in the frontal lobe that are involved in the processing of faces. In particular, these areas become active when we see a face and then must hold that face in mind when it's no longer present in order to recognize it a short time later. Studies such as this tell us that areas of the frontal lobe are involved in maintaining these short-term memories for faces. We also have evidence that the very same areas of cortex that are involved in the processing of information about faces also participate in the long-term storage of memories for faces for subsequent recollection. In fact, when we imagine a face in our mind's eye, these very same areas of cortex also become active.
Higher vision is not limited to faces. It may help pick out the one lame animal in a herd of 25,000. For the lion, such visual computations are everyday tasks. Does he, could he, meticulously examine all 100,000 legs for wounds? Or does he look for visual exceptions, for an irregularity of movement that stands out more clearly than a bloodied leg? How do we spot an old friend walking away from us in a crowd? By the color of his socks or by the way he moves? Either way, while we call this process seeing, it's a very high order of seeing. It's perception. And how long does it all take to happen? Well, the process of seeing and perceiving something as elementary as a flash of light takes about 3/10 of a second. There, that's vision. Blink your eyes and you might have missed it. Slowed down, the route of vision is clearer. First, we can see the back of the brain light up, here in the area studied by Hubel, Wiesel, and de Valois. Now, with images being computed along those pathways of Mortimer Mishkin, the collective information reaches the front of the brain, the thinking part of the brain. And it's now when we realize what we've just seen. For another clue to the brain's remarkable adaptability, consider Susanna Fiennes, a London art student who has agreed to undergo a curious experience. In order to appreciate this, we need to review how the visual mechanism operates in our normal everyday encounters. For example, when an object appears in our visual world, the image passes through the lens of the eye, and in that process it is inverted onto the retina. The image is then passed along the visual pathway to the visual cortex at the back of the brain, where it is perceived as being right side up.
Susanna, if I could have a look at these most unusual spectacles on you.
For one whole week, she will see the world upside down, inverting once again what the lens in the eye does anyway. Our brains correct for this inversion. Now hers will be forced to make yet another correction. Will she be able to make sense of an upside-down world?
Can't be.